Hello and welcome to the University of York Department of Archaeology Prospective Postgraduate Studies Seminar Series. Each episode features some of the fantastic staff we have here in the department. We are putting this series together to give prospective students a sense of who they will be learning from, what kind of research is happening, and highlight what makes our department a uniquely wonderful place to undertake a master's degree. In this first episode, we are going to hear about some of the incredible work in funerary archaeology, zooarchaeology, and bioarchaeology. Enjoy!
My name is Dr. Penny Bickle. I am the Director of Studies for the Funerary Archaeology programme. And as you know, we have an MA and an MSc route. And over the next 15 minutes or so, I want to tell you a little bit more about why we have those two routes through giving you a little bit of a history about how I myself became a funerary archaeologist. So I specialise in the Neolithic period. So that's when the first farming communities spread across Europe. What I find so fascinating about the Neolithic period is its mix of familiar and unfamiliar. On the one hand, much about the Neolithic period seems familiar to us. They ate food that's familiar, such as wheat and cereals, and animals such as cows, sheep and goat and pigs. They lived in villages and in houses that are not far away from what many people live in today. Yet there are hints that their worldviews and their cosmologies were very different to the ones that we have today. The way they treated the dead, the monuments that they built, suggest they interacted with their environments in really different ways. And it was that juxtaposition between the familiar and the unfamiliar that enticed me into doing a PhD thinking about Neolithic settlements and houses. So why am I now a funerary archaeologist? How did I go from doing my PhD, which I finished in 2008 studying Neolithic longhouses, to directing a funerary archaeology programme today? Well, when I started my PhD, I would be surprised, as you probably are. I was really interested in the rituals of daily life and how those shaped the spaces that people lived in. So I was super interested in houses and their layouts and what they were like to live in. And there's a picture of me here standing proudly just before my dissertation was finished in a reconstructed longhouse in Germany. However, as I did my PhD, I became more and more aware that if I wanted to study the Neolithic period, and particularly the houses, I needed to think about how death was integrated in that. And I was really lucky that after my PhD, I went on to work on various projects working with human remains, particularly working with isotopes. So here I am in Austria sampling human remains for dietary isotopes, so carbon and nitrogen, to look at the kind of proteins people were eating. And then as I've moved on with my career and I had more projects, here I am in 2018. I'm in actually a library in the Museum of Archaeology in Strasbourg, which is the only space they had for us to do some sampling. And I'm with one of my postdocs, not pictured here, doing some analysis on the skeletons. And in fact, here we were visiting the collections to take some dental calculus, dental plaque samples, which we're still working on at the moment. So it was really settlement burials and kind of thinking about settlements that got me into thinking about funerary archaeology. So in the Neolithic, across many different areas of Europe, we find burials alongside houses. They're in the pits associated with houses, they're under the floors of houses, they're even found in disarticulated states alongside and around in different pits. So I was focusing my PhD on the Central European Neolithic, where we have these longhouses. And when we think about them, when we reconstruct them, we often look at this. So this is a lovely reconstructed house that comes from an open door museum in Austria called Mammuts. And I was very lucky to get a chance to visit with my colleague Eva Linnes. And as we were walking around these beautiful, pristine houses, we were talking about what it would really be like to have visited one in the LBK. And we think it probably would have been more like this, in fact. There's lots of pits, lots of waste open on the settlement. Distributed all over around the walking surfaces. We find bits of pot and bone distributed everywhere. And when I was doing my PhD, the real assumption had been that actually where we found disarticulated human remains in these rubbish pits and burials on settlements, they were low status, they weren't very important, they were being incorporated into this rubbish. perhaps because it wasn't very important and they were low status. However, while I was doing my PhD, those days were a bit more analogue, and I had to go to the libraries at the University of Oxford, where they published a whole series of excavations, and I was working through them, huddled away in the library looking through. I came across this burial here. So this burial here is a child burial, and it is someone who probably died about the ages of 4 to 6. And rather than being thrown out the rubbish or just incorporated into the waste of the pit, someone had made time and space to clean out a little scoop within the daily waste, and they'd sprinkled that scoop with ochre, and that's the red dots that you can see on the screen here, and then they'd nestled the child within it. And as I was kind of reading the archaeologist's description of this excavation, it really struck me. This wasn't a low status burial. This wasn't someone being thrown out with the rubbish. Care and attention had been given to this child to really make sure that they were safe as they were nestled there, perhaps for the afterlife, or just perhaps to be kept close to the house and the people who were mourning them themselves. And so I argued that rather than rubbish informing how we thought about the dead on Neolithic settlements, we should use the dead to think about how rubbish had a different cultural response in the Neolithic. So rather than being something which is devalued, not very important, perhaps it was very important, something about keeping life around you, keeping those material remains close. And this care and attention that was taken to these burials was not neglectful or low status. These were people using their own environments to mourn the dead in the way that they saw most appropriate. Since I finished my PhD, more evidence has come to light that suggests that Neolithic people were not necessarily just burying people in single inhumations and then forgetting about the burials. We have numerous evidence that graves are returned to, bones are removed, and in fact in some cases where very careful excavation has taken place, we find extremities, so fingers and toe bones, suggesting that the body had been placed in the pit just to decay, with it later being revisited and the bones removed. So this is something that's a very different approach to death than we experience in our own modern world. So what do we think's going on here? Well, we have various ethnographic examples of places around the world where people choose to engage with dead bodies long after the deceased has passed away. I've picked out 3 examples here that I find particularly interesting for studying death and burial in prehistory. So in Imani, in Greece, until recently, there was a practice of once the bones had been cleaned and being moved into a temple or moved into some kind of charnel house, the bones would be looked over for divination. It was a time of great emotion, but also comfort for the mourners as they looked after the bones of their relatives. Looking more further afield from where we are here in York, we have practices such as the Famadia in Madagascar. This is called the turning of the bone ceremony, where ancestors are revisited at particular ceremonies. The bodies are removed, cared for, rewrapped. Children who have been born since the person passed away introduced, stories told about the way that they lived. And then finally here, the Manena festival in Toraja in Indonesia. In Indonesia, people are not said to be dead when they first die, only when the funeral takes place. So this means that for many relatives, they may stay in the house after they've passed away, with people saying that they're sleeping or sick and still offering them food and including them in activities in the household. What do I think is going on with these particular practices? Well, I think this is about having a drawn-out relationship with the dead. The way we handle death in the modern world, of an instant disconnection, medically looked after and then handled by specialists, is very different from how people would have approached death in prehistory, where you would have cared for your relatives, and perhaps this was a way, and perhaps even a healthier way, of mourning the loss of that particular individual. So since the end of my PhD, I've been working on projects which bring together funerary archaeology and daily life. Lifeways and deathways, I like to say. So what I'd like to do now is to turn to some of my current projects and give you some exclusive previews, as we haven't published any of this data yet. So Our project earlier, I was talking about settlement burials, led us to think a little bit more about cemetery burials. So settlement burials had been considered low status because we also found cemetery burials in this early Neolithic Central European culture. But why? Why would you have both settlement and cemetery burials? So cemetery burials have been considered high status, I would argue, because they are the norm in our own culture. Settlement burials actually have a much longer history in the Neolithic and continue throughout, whereas cemetery burials only seem to start about 200 years after the first spread of Neolithic communities. So we wanted to know more about this spread. Were cemeteries a new fashion that spread with increased hierarchies? Someone was doing it down the road, you wanted to be a high status person, so you started a cemetery? Or was there more things going on here? So we started a project dating Neolithic cemeteries to try and work out in a little bit more detail what was going on with these cemeteries. We were really lucky to get quite a lot of radiocarbon dates, and what we found is super interesting. So rather than cemeteries being a fashion which spreads gradually, they seem to start really, really quickly. And most of the cemeteries we dated do not last a very long time. So rather than sort of lasting centuries and centuries, we see 2 to 3 generations of burial, at most 4 generations of burial, so about 80 to 100 years at the very most. So this is really different. What could possibly be going on here? So after coming out of COVID, it's really hard not to speculate that this is perhaps a specific response to an increase in the numbers of people dying, perhaps some kind of pandemic or outbreak. Perhaps it was also related to an increase in population. Today, we're really struggling with the energy costs, but also the space that burial takes up, and communities around the world are having to face choices about what they do with their dead now if cemeteries are filling up. So here we might see the same kind of thing happening in the Neolithic, very relevant for the issues that we're thinking about today. Cemeteries were not then some kind of high status way of being buried that came in and then continued or evolved out of settlement burials. It was something that happened very quick. Explaining why is a little bit more complicated and just sets us more research questions to consider over the next few years. So this knowledge about cemeteries led me to be curious about who was being buried at settlements and cemeteries and whether there were any differences. And I was really lucky a couple of years ago to be contacted by a team who were working on the A DNA of the Neolithic. They had a new, what they call genome-wide approach, which allows us to look at biological relatives being buried at the same sites. So we can see biological relatives up to about 3rd degree, so cousins. Together with the project, we selected 3 sites to compare from the early Neolithic: a settlement, and its settlement burials, the burials alongside the houses, a cemetery, and then what we call a mass burial site. So something that we thought might represent a living population. This site is called Aschbarnschletz. It's got about 78 people who appear to have been died by the hand of some kind of violent activity. They're disposed of in not very ceremoniously ways in an enclosure ditch outside the settlement. And we hypothesised this was the population within the settlement, or at least it's been hypothesised like that for a long time. They were living together, a raid took place, and then the survivors buried the dead very quickly around the enclosure. So this was our living population, that was how people were living, and we were going to be able to compare that to who was being chosen to be buried on settlements and who was being chosen to be buried in a cemetery. So what did we find? Well, at our settlement site, we found 3 large family groups who were buried close together in those family groups. So kind of what we'd expected. Alongside the houses perhaps they'd lived in, 3 big groups of people, a little bit of sort of coordination between the two, but otherwise sort of lots of relations, grandmothers and aunts and uncles and cousins and things coming together on those settlements. The cemetery showed us something a little bit different. Rather than having lots of big family groups, we actually had small clusters of perhaps 2 to 3 relatives. which again speaks to something that's a little bit quicker in time than something that builds up over, say, 400 or 500 years. And one of my favourite burials is a weird thing to say, but one of the ones I found super interesting was this triple burial. Here in an earlier project, my colleague Linda Fibiger, an osteologist, had identified that the two small children buried here, had died by blows to the head, and they'd been placed with an adult female. So it had long been speculated this adult female was their mother, but when we did the DNA evidence, we found that she was in fact their paternal aunt. So a real indication that this isn't just about nuclear families. There's all sorts of different kin relationships taking place across these different sites. But then we hit on a real mystery. The massacre site, 78 people, we only had 2 people related. And so that left us a bit of a quandary for our interpretation. What was going on at this site? Do we have a central site where people have come during a time of conflict? Do we have actually some kind of sacrifice or tribute taking place? It's an open question, I think, and again, more questions set for us to research into the future. So overall then, I would argue that funerary archaeology, both the MA, Humanities, Social and Cultural Practices, and the MSc, the science, the analysis of the human skeleton, need to come together for us to investigate the ways in which Neolithic people approached death. And funerary archaeology here is providing us with a really exciting window into some of those different worlds that Neolithic cultures lived in. Thank you very much for listening to me today, and I'm really happy to take questions. Thank you very much.
I'm David Orton, and I run the MSc in Zooarchaeology here at York. For this seminar, I want to talk about a new research project on rats, which I'm personally very excited about, but which I also think is a really good showcase for the range of methods that we use here in Bioarch. As animals go, you might think that rats don't really require a lot of research. After all, they're already only too familiar to humans all over the world. But what do we really know about rats? They're a pest, most people would say. They live around humans, they raid our rubbish, spread disease perhaps, but where did these furry little camp followers actually come from? How did they end up on every continent apart from Antarctica, and how long have they been with us here in Europe? For that matter, is their reputation as plague spreaders deserved? The Rattus project aims to address all these questions, amongst others. Rattus is a new project based in Bioarch at the University of York and funded by the UKRI. Over the next 5 years, we'll be aiming to understand the intertwined histories of rats and humans in Europe across 2,500 years, from the Iron Age to the 19th century. The basic idea here is that if rats are like a shadow to human societies, depending on human settlements for food and shelter, and on human trade routes to get about, thriving where we thrive, then we can use them as a proxy for broad trends in human economic history, themes like trade, urbanism, and migration, which feature heavily in both prehistoric and historical archaeology. At the same time, we need to understand the impacts that rat populations may have had on humans, most obviously through disease. The project is based around zooarchaeology, the study of archaeological animal remains, in this case tiny rat bones recovered from hundreds of sites across Europe. But the project also includes all the advanced biomolecular techniques for which Bioarch at York is known. Ancient DNA to track routes of dispersal and connections between regions, stable isotopes to understand changing dietary niches in different types of settlement, and of course protein analysis in the form of zooms, a molecular identification method invented at York about a decade ago. We also have experts from the history and biology departments on the team, bringing skills in the analysis of written documents and in ecological modelling techniques respectively. So, when did rats reach Europe? Well, it depends what species we're talking about. The rats that you're most likely to be familiar with today, hopefully not too familiar, are brown rats, Rattus norvegicus. These are probably originally from northern China, southeast Siberia, maybe Mongolia, and likely didn't arrive in Europe until the 18th century. Previously, the only rat species in Europe was the black rat, Rattus rattus. This is the species linked to the Black Death, and it actually comes from South Asia, so it's a long way beyond its native range, and a long way beyond its native climate here in temperate Europe. This makes it typically dependent on human settlements to some extent, and therefore very useful for archaeology. Research on the genetics of modern rats has given us a rough global picture of their spread, but not the timing of that spread. We know that black rat reached the Mediterranean by the Roman period, but we don't know much more than that. Rival theories exist, but no one has yet tested them. Rattus will track down early reported finds of rat bones from pre-Roman sites and check them with zooms and with radiocarbon dating. Next, we want to understand rats' dependence on human settlements and economies. The Roman period is a great starting point here. This map shows rat finds during the first half of the first millennium AD, and as you can see, the rat finds basically form a map of the Roman Empire. So why is it that we find rats all over the Roman world, from Egypt to Hadrian's Wall, but almost never beyond the frontier? The easy answer would be that rats thrive in cities, but it's not that simple. We also find them in forts and in villas, small but well-connected sites. I suspect that rats were dependent, on a metapopulation level, on the Roman economic system as a whole, with its combination of large urban hubs, smaller but well-connected settlements like villas, and most importantly of all, regular bulk trade in perishable goods like grain. This included commercial trade, but also state-organised provisioning of major urban centres and, famously, of the military. Rattus will look at this systematically, mapping out finds by settlement type, assessing connectivity with ADNA and dietary niches with isotopes, and building population models of the whole system in order to understand what aspects were most crucial to sustaining rat populations over the long term. But if rats were dependent on the Roman economic system, what happened when that system broke down as the Western Roman Empire started to collapse from, arguably, the 4th and 5th centuries? Well, the rats seem to have gone with it, at least in northern provinces like Britain. This broad pattern has been known since the 1990s, but Rattus is firming it up and refining it, checking anomalous finds, improving the resolution of our dates, and also looking in regions which have previously had relatively little research. Already we can show that this decline happened in the 5th century in England, but not until the 7th century in Italy, suggesting that it really is down to urbanism and economics, not, for example, to the plague and climate events of the 6th century. This lends some support to arguments for a sharp change in economic conditions as the Western Roman Empire fell apart, at least in provinces like Britain, a point which is widely debated by historians. A recent genetic study by Rattus team members and others seems to back this up. We showed that Roman rats all across Europe were closely related to each other and almost completely replaced by a new population in the medieval period. That leads on to another big question. When and how did rats repopulate northern Europe? Some of the earliest reported medieval finds are surprisingly far north, including the Viking Age trading sites at Birka in Sweden, Hellebu in northern Germany, and here in York, all in the 9th century. These finds emphasise the importance of a network of trading sites around the North Sea and Baltic that had been developing since at least the 8th century, the Northern Emporia. But what was it that changed in the 9th century and allowed rats to colonise key nodes in this network? Possibly their increasing size and urban character, but we also need to think about connections to other regions and where the rats might have come from. Did rats reach the North Sea through the expanding Carolingian Empire and spread east, or did they reach the Baltic via the early Rus river trade with the Byzantine and Islamic worlds and then spread west to the North Sea? We will build on the previous genetic study to test these hypotheses, while also looking for early finds in the currently understudied Eastern Baltic region. By the 13th century, rats seem to be well established across most of Europe, and it's against this backdrop that the Black Death famously devastated the continent in the 14th century. Black rats are firmly embedded as the villains in this pandemic, both in the popular imagination and in traditional academic narratives, but in recent decades there's been a move to exonerate rats, in favour of other mechanisms for the spread of the plague bacterium, such as human lice. Without taking sides prematurely, our project hopes to advance this debate via a better understanding of the rat populations themselves. Were rats present in rural settlements or only in towns and ports? What about in the far north? We'll systematically compile data from across the continent, not just where rats are present, but also where they aren't, but other small rodents like mice and voles are. We'll model the likely population dynamics of rats, starting at individual settlements and building up to a continental scale, informed by our zooarchaeological and archaeogenetic results. And we'll also look at the impact that plague might have had on rats themselves, both via their frequencies through time and via any genetic evidence for selection. If we're lucky, we might even pick up plague bacterial DNA from infected rats. Finally, we'll look at how the black rat's dominance in Europe came to an end. We'll use zooarchaeology, zooms, radiocarbon, genetics and historical sources to chart the spread of the brown rat, Rattus norvegicus. Based on written sources alone, we can already rule out the most popular narrative in modern textbooks, as spread through Russia and Central Asia, and replace it with a possible Caucasian route, while the maritime dispersal to Western Europe with colonial shipping is also very possible. What impact did this new species have on the existing black rat population? We'll assess this through written accounts, through genetics, and also by looking for stable isotope evidence of changing dietary niches for the black rat. It's an ambitious project, but we think we can make a major contribution, not only to the history of rats, but to the history of humans too. There's a lot of work to be done, and we're already involving students here in York in the research. It's going to be an exciting 5 years.
Hi everyone, welcome to this session on bioarchaeology. My name is Lara Gonzalez Carretero. I'm one of the lecturers at the department. I'm a lecturer in bioarchaeology and in archaeobotany. That is the study of plant remains from archaeological sites. And I focus my research on the analysis of food remains. I'm going to talk a bit about this, but before that, I'd like to say a bit about my previous research before joining University of York. I joined this year, actually, last year, 2022, in September. My previous research focuses on past diet and cuisine, and specifically on fruit procurement and agricultural practices in Southwest Asia. However, I had the chance to work in commercial archaeology for the last 4 years, and that allowed me to gain a lot of experience and knowledge on British archaeobotany. I am at the moment in the Department of Archaeology in York. I'm part of the Bioarch facility, and we are a very varied group of specialists who actually apply the latest scientific techniques to the study of biological remains and archaeological artifacts. We apply the cutting-edge technology such as isotopic analysis, lipid analysis, ancient DNA, proteomics, etc. to the study of plant remains, animal remains, and human remains mostly. But we also apply these techniques to artifacts such as pottery, groundstones, etc. I, as I said, focus on the analysis of archaeological food remains, amongst other things that I'm interested in. This is my main focal point of research. And when I say food remains, I mean these things you have on the screen. There are two main types. So the first type would be something like the ones you have on that petri dish in that picture. So these are isolated food lumps. We call them charred food remains, charred food fragments, things like that. These look like, really they don't look like much. They look a bit like charcoal. But in reality, these are the remains of meals such as breads, porridges, and things like that people cooked in the past, and they got preserved through charring and burning. And they get recovered through the flotation of the soil when we excavate archaeological sites. The second type that is very commonly recovered is food crusts. So food crusts, as you know, the name indicates, are crusts of food that were formed through burning while cooking, and they were preserved in the inside of pottery vessels and ceramic containers. And we are able to identify, what people were cooking through the analysis of these things. I had the chance to develop the new methods for the analysis of food remains, and I did this on focusing on samples recovered from one of the most important sites in the world. This is the site of Çatalhöyük. I'm sure you've heard of it because it's it's an amazing site. It's a UNESCO site. It is located in central Anatolia, and it has such a good preservation that you can actually see the buildings perfectly preserved with all their features, separations, etc. You can see that on that picture there. And as I said, it's in Turkey, and it is one of the longest occupied sites in the world. This one has, in particular, a very long occupation of more than 1,000 years, from 7,100 BC to 5,950 BC. So as you can see, lots of opportunities here for us to study the diachronic use of plant remains and archaeological food techniques, etc. My methods focus on the use of scanning electron microscopy, or SEM. The way that I apply this to the study of food remains is that we do take these food remains and put them under the scanning electron microscope, and we can see with high resolution images, we can see the ingredients and things that people were using for the preparation of these foods, as well as how these things were prepared. So depending on the type of microstructure that we see, so if we see lots of air bubbles, these are probably things that have been boiled, but if we don't see this type of air bubbles, then these things are probably being baked, etc. So we can see differences under the scanning electron microscope that can lead us to to take a conclusion on how these things were cooked. I also apply experimental archaeology to my methods. So I try to replicate the meals and the recipes and the things that these people were cooking in order to compare with the archaeological samples that we have and interpret what people were actually eating. And I focused on the preparation of 3 main meals. So these are type of breads, flat breads, but also leavened breads, and doughs like dough balls or bread doughs, and porridges or gruel type things. My methodology and my research actually led to the discovery of the oldest bread in the world. This bread was discovered from the site of Shubeika 1 in Jordan, and it's a almost 15,000-year-old bread. We were able to identify little pieces of bread, up to like 40 or 50 pieces, I think it was. And we actually identified that these things were not only made with wild cereals, because this discovery is so early on, is before cereals were domesticated, before we have cultivation and agriculture, extensive agriculture all over the world. So these things were actually made with wild cereals that people gathered. But not only wild cereals, we were able to identify that actually people were also gathering something else and mixing it with cereals. So it's not a pure cereal bread, it's also made with a different plant as such that people were recovering, but instead of using the leaves or the stems of this plant, people were actually using the tuber. So that's the rhizome or the root of the plant, and they were grinding this and they were mixing it with the cereal flour. We carried out experimentation, as you can see here on the screen. Again, we gathered the wild cereals, we gathered the tubers from this sedge that I mentioned, we ground it, and we made a flatbread, and we baked this on the hot ashes of a hearth, and also on a slab on stone, to see which cooking technique was more similar to the one that people could have been using in the past, because these people didn't have ovens yet. That was quite fascinating to see, actually. And our discovery actually changed everything in archaeobotany. Our discovery changed the way we looked at cooking and bread making in this case. Until now, we thought that bread was associated with the Neolithic communities and with people who already had extensive agriculture and cultivation of cereals, and that cereals were the staple food for them. However, we were able to see that bread was actually something made very early on, before agriculture was established and before cereals were domesticated. And it was worth it for these people, for some reason, to gather wild resources and invest their time into making a bread. My methods actually also helped for another discovery. This one a bit closer to home, to here. So this was the discovery of one of the oldest remains, physical remains of beer making. These remains that you can see on the screen were recovered through the excavation and processing of soil from the A14 road scheme. It was led by Highways England and excavated by Mola Headland Archaeology. And I was one of the archaeobotanists who were able to, study these remains. And through the application of my methods, I was able to identify that these remains are probably derived from the mashing stage in the brewing process, in the beer making process. I also wanted to say a bit about a project that we are currently working on. This project is a European Research Council project, and it's in collaboration with the British Museum and Kiel University in Germany. It is the Induce project, and it focuses on the study of hunter-gatherer pottery from Northeast Europe. We had the chance to analyze more than 800 pottery sherds, and I myself analyzed something like 150 food crust samples. And it was very interesting to see how plant remains were present in most of them, and in particular these berries from the plant of the Gelder rose, Viburnum opulus, and these berries are actually toxic. So it's really interesting to see how people actually chose something that is not considered a food again to put in their pots and cook and prepare some type of meal with them. We also applied and combined the microscopy that I focus on with biomolecular analysis or organic residue analysis. These are, as I said earlier, isotopes and lipid analysis. And thanks to the combination of the two things, we were able to also identify a range of animal products that people were using to prepare their foods with these pottery vessels. In particular, the use of aquatic animals or fish, freshwater fish in particular, but also a range of other things such as ruminant fats, dairy, etc. Something that we were also very interested in was the application of experimental cooking to the whole project and to the comparison with the archaeological remains of food. So we chose ingredients that were identified through my analysis and lipid analysis, and we did research into ethnographic cooking in this area of the world, and we came up with a range of recipes that were probably those that people were trying to cook or cooking in the past. So we tried to replicate this through the experimental cooking, and we carried out a set of experiments on an open fire, as you can see here on the screen. And we were also able to taste some of these recipes, and I have to say some of them are not very tasty to our senses today. But again, we have changed so much so that who knows how these people actually, experienced food. And this project led to the conclusion that cuisine is actually socially situated, of course, and some of the things that we see in these pots that have been used to be to make food are not necessarily reflective of subsistence practices, such as like the Gelder roseberry, which is not actually a food. So it doesn't really represent the daily staple foods that people would eat, but it is more a social and cultural attribute rather than an economic indicator. I just wanted to say thank you as well to all my collaborators. And thank you to the grants and the funding that I've had during these years. And this is the end of my presentation. If you have any questions, please feel free to email me. And if you have any questions about the program, about the MSc in Bioarchaeology, please contact us. We are more than happy to help. Thank you.
Hello, I'm Dr. Ashley Lingle. I am a lecturer here in the Department of Archaeology at York, and I am also the PGT or Master's Admissions Lead. So hopefully you've really enjoyed the talks that my colleagues gave ahead of this presentation, and I thought what I would do now is just give you a little bit of a flavor of what it's like to undertake PGT or Master's study here at the University of York in our lovely archaeology department. So, what's it like? We have a really diverse range of taught degrees, a very dynamic teaching team, which you'll see hopefully from the presentations you've just watched, but throughout this seminar series as we highlight some of our incredible staff and the degree programs that they teach on. You'll see that it's really cutting edge research, but it's also pragmatic, so there's a lot of professional applications. While you're here, you'll be learning independent study skills and how you can contribute within the field. And I really can't stress enough that we've got a really diverse team that are influential thought leaders, and they're really leading the way in what's being done and studied within the scope of cultural heritage. Our teaching approaches are diverse, so we use a broad range of assessments. We really like to get you out there doing field trips, kind of across York, but other parts of the UK as well. And then in the classroom, a lot of seminars, discussions, group work, and then a variety of assessments as well. So you get a sense of not just kind of formal learning structures, but really how to collaborate. We have a very broad range of courses. Again, you'll be able to see these as our YouTube series develops. But we've got a range of both MA and MSc degrees. So those of you interested in science or less science, but across different time periods, material types, etc. And again, a number of heritage degrees, so not just archaeology, but cultural heritage management, conservation, and a new museum studies program coming up. We also share sustainability studies with our Department of Environment and Geography. While you're here, the degree structure, you'll take 3 20-credit modules in your first semester, and then again in your second semester. And some will be core, but then you'll also have some optional modules depending on which program you're selecting. And from there, you'll do an 80 or 60 credit dissertation depending on whether or not you're involved with some of our placement programs. So here's a very long list of modules in semester 1. Again, you'll have some that are core depending on your program, but you'll also have some optionality, and so you can really tailor it to your interests and what you would like your future career to look like. And again, in semester 2, a real wide variety of options, including the curated placements where you'll be working with different partners throughout the UK to undertake kind of very practically based work. If you're interested and you're not sure how to pay for it, we've got some funding support that you can access, as well as access to your individual directors of study. So if you know what program you want to apply to, go online, have a look at who you need to contact, and just have a chat with them. Once you're ready to submit your application, have your academic transcript 2 pieces of written work. These can be essays or professional reports, anything like that, just to give us a sense of your writing skills. Your CV, if you need a language certificate, a personal statement, and then some references. It's all managed through the online portal, and it's very intuitive. So again, if you're interested, please have a look at the different degree pages and contact your directors of study. For the program that you just watched, please have a look at these 3 individuals as they're the lead directors of study for those programs. Thank you very much, and we hope to see you here at York Archaeology.